So this is the last module of the Unit 4, Module 8, which is the cancer cell biology. And you will see that many of the chapters that we already talked about, they will all come together and make sense. And we will just go about almost like a 360 degrees in terms of explaining that how all these things actually work in concert. So you will see that the very first cell culture experiment, that's something we talked about. And then we just learned in the cell junctions and right here, when we talked about the benign and the metastatic tumors, how these things actually come into play, non-functioning of these things come into play. Then we're going to talk about that the cell cycle that we talked about in the previous in Unit 3, how the dysregulation of that can lead to these cancer. All right. So first part is the general consideration where we are looking at some of these facts that these are very self-explanatory. Just remember that the tumor cell is sometimes referred to as transformed cells. Sometimes they are called neoplastic cells, just neoplastic cells. So where it's acquired neoplasticity means it's just uncontrolled, unregulated fashion. Sometimes neoplastic transformed cells or just neoplastic cells, cancer cells, these are interchangeably used in that. All right. So basically, the idea is that the cells grow and divide in an unregulated fashion. And it is the breakdown of the cell's exquisite control of regulating the cell multiplication. So essentially, the disruption of the cell cycle control, what we are going to see that the tumor not always means that it is a cancerous tumor. And we're going to see that what are the differences between that. But anything tumor is an uncontrolled cell growth. So we'll just come to this, which is a benign and malignant tumor. And immediately, these are your important questions that can come from here. So here we are looking at that a benign tumor, which becomes localized. Usually if they become dangerous, bulky, biologically active, sometimes if they actually secrete some of the hormone, which impairs the normal physiological activity of the organism or the person, then it may need to be surgical removal. That's basically the idea. But just think about that. A benign tumor could be as simple as a skin tag, that the skin tag is also a benign tumor that's essentially an uncontrolled cell growth. Now, the benign tumors are different from the malignant tumors, that malignant tumors grow and divide more rapidly than the benign tumors, and they actually move from their own territory or the tissues into the other area, which are normally reserved for other cells. So you can easily see that here you are looking at these cells. Here the cells are pretty much connected with a fibrous connective tissue, and they are localized. And here we are talking about something which we call as an adenoma. But when we are talking about it is becoming an adenocarcinoma, that means it is, that adenocarcinoma is a malignant or metastatic tumor which can invade to the other spaces. And these cells are aberrant, means that they do have different, sometimes shows very different morphology than the original cells as well. So what type of question that we can ask? So we can ask which of the following is a characteristic of a benign tumor, which of the following is not a characteristic of a benign tumor. The same way we can ask that which are the following characteristic of a malignant or metastatic tumor, or which of the following is not a character of metastatic tumor. So let's see an example. Let's suppose if we say that which of the following is not a characteristic of a benign tumor. So the main characteristic of a benign tumor, which is not a characteristic of benign tumor, will be that it is not malignant or it is not metastatic. So if you see a benign tumor, we are going to see that they are usually covered in a fibrous tissue, which is this fibrous connective tissue. So I'm just pointing it down right here, fibrous connective tissue, and we can see that there will be a layer of a particular material which we call as a fibronectin. This is fibronectin. So definitely write this one down. We are going to see that word in a slide just after this. So it is there. So, but just connect that as well. The fibronectin. When we talked about the integrin in the previous chapter, you saw that the integrin, you have this alpha and the beta. and the beta chains. And then there was a picture that something is connecting to that. And so this is your integrin. This is one of the cell adhesion molecules, integrin. And the integrin actually binds to one of these fibronectin, which is one of your ECM proteins. This is your ECM protein, fibronectin. And the cells actually bind to it. And here you can see this fibrous connective tissue that contains the fibronectin. The cells are attached to with the ECM in there, not going anywhere. But that coat of the fibronectin is reduced in the malignant tumor. We are going to come to that. So that's one of the important things. But if we just have a choice that which of the following is a characteristic of a benign tumor, they are covered by this fibrous connective tissue rich in fibronectin, and that is correct. But if we just have a choice that which of the following are not a characteristic of benign tumor, remember that your malignancy or metastasis is not a characteristic of that. They do not colonize other territories. These are usually not the characteristic. On the other hand, just remember that the same way that if we just ask is which of the following is a character of the malignant tumor, remember that will be the metastasis. So if we ask a question which is not a characteristic of a benign tumor, this will be the answer. On the other hand, if we just say without making it a negative question, which of the following is a characteristic of a tumor, this could be the answer as well. So just read the question, whether the question is asked with a not or which of the following is a character. So that's basically the idea. So when we see on the other hand, when we talk about the malignant or metastatic tumor, if we just say that they are well differentiated, that will be incorrect. They are not well differentiated. They have all these differentiations are just like very aberrant. They are not well differentiated. And if we just say that they sense the signal that restrict cell division, remember that both of the benign as well as malignant tumor, they cannot sense the signal for restrict the cell division, because this is an uncontrolled cell division. So if we just see that a malignant or metastatic tumor cells can sense a signal that restricts cell division, that will be incorrect. So these are some of the options or choices that we can have in those areas. So, all right, so let's move on to the next part. These are the cells in culture. So you have seen that pretty much the first few slides of your unit 1, we talked about that you need essential amino acid, vitamins, growth factor, require solid surface, anchorage dependence, display contact mediated inhibition of growth. And we are going to see that you do not need this slide particularly directly. That's not a question mark, so right here. But you can see that this anchorage dependence and contact mediated inhibition. These are the two characteristics of a normal cell. Remember that. And what we are going to see that these properties are going to go away when the cells become cancerous cells or malignant cells or neoplastic cells. So these properties these are the properties of normal cells in culture, and these properties are going to go away. The anchorage dependence, contact mediated inhibition. So what happens is that when you have the cells, and if a cell while growing touch each other, they will stop growing. But what happens is that when you have the cancerous cells, they will pile upon each other, and they do not care about these contact mediated inhibition as well. And anchorage dependence means that at the bottom of the Petri plate, they just attach to themselves to the bottom of the Petri plate, because they secrete these different ECM proteins that will basically attach on the etched surface of the Petri plate. And then they will have all these integrins that will attach them onto the ECM proteins. Now, what happens is that when they lose this control, they essentially lose these ECM proteins, and they are not attached to them, and they just come loose. They can just stay in the bottom, but they just come loose, and they usually lose their anchorage dependence. And we're going to see that in the next slide. That is more important. So here we are looking at that you have the normal cells of these cells. You can see that they are towards the bottom of the cell in this scanning electron micrograph, whereas here you can see that transformed cells, they are piled upon each other. So you can see that they have changed. See here, they are more of a flattened, elongated cells. Here, some of them are round, and those, so morphologically, they are a little bit different as well. So this actually tells you that a normal cell have contact inhibited monolayer of normal cell. But here you have a plastic tissue culture dish where you have this multilayer of uninhibited cancer cells. So when we see in a batch of normal cells that we are growing, sometimes if we just add some mutagen or any cancer-causing agent, we can transform some of these cells in the media. And we are going to see that there are some of the areas where there is a focus of cells that actually pile upon each other, and we call that as a focus or foci in a plural of cells, which is a piling upon the cells upon each other, from which we know that they are the cancerous cells. All right. So you can see that there is a lot of underlining in here, and there is a reason for that. You can expect multiple questions from this slide and the slide after this. So both of these are very important slides. So just make sure that you get all of these religiously know this. So here, what we are looking at, that in the transformed culture cells, first, we are looking at some of the adherence abnormalities. And the second part, we are going to see that what are the growth and the division abnormalities, and just know all of them very, very well as well. So first, we are looking at that the diminished adhesion to the surface, therefore able to maintain a rounded morphology, and see that if you just look at that there is a couple of cells still they are flattened, and that is almost a normal morphology. Now the cells that are on top of them, they have just become rounded. And this is what we were talking about, that the cells, when they become transformed cells, they sometimes show this aberrant morphology, which is different from the original. So they are not well differentiated. So these are well differentiated, only one type of cell. Say if we just go to the picture right here, these are all well differentiated, one type of cell. But here you are seeing that they are rounded up and they are aberrant rounded morphology. All right. So the second one, what you see is the adherence abnormality, the failure of actin filaments to organize into stress fibers. So what happens that these cells are flattened, and their cells are actually kept in that place because you have the stress fiber. If you remember that if you have a cell like this, the stress fibers run across the cell straight, and this actually keeps it. So here is the nucleus of the cell, and it just runs across, and these are your stress fibers, which allows the morphology of the cells. These are not, again, remember that we are not talking about the lamellipodia based actin that is modeled on the lamellipodia on a moving cell. This is a cell which is just a static cell, and these stress fibers actually keep the shape of that, and these stress fibers are not properly organized in these cases, and that's why they just become this rounded morphology instead of see that this is a flattened morphology at the bottom. They still probably have some stress fibers going along with them, but see that this rounded morphology means that they do not have a proper actin distribution, and that's why that problem is happening. After that, we can see that reduced external coat of fibronectin. So this is actually very important. Remember that reduced external coat of fibronectin, because remember that the fibronectin is a ECM protein that is secreted by the cell, and the integrins actually bind to that. Suppose this is a cell that you actually transform that cell. The cell will actually secrete this fibronectin or produce this fibronectin that will cover the surface of this plastic. And then the cells will extend their integrin and bind to them and anchor them. So this is the anchorage dependence. But what happens that when they have the reduced external coat of fibronectin, when they cannot release these ECM proteins, remember that fibronectin is one of your ECM protein. And when they cannot reduce these ECM proteins, that means that, see that right here, the blue surface, this should be covered by the fibronectin, which is becoming less and less. So the cells will not be able to adhere to them properly. And that's why they lose their anchorage dependence. So when we look at the main, one of the important thing in here, the anchorage dependence, because remember that anchorage dependence is required, aided by the fibronectin. That's important. All right. So after that, the number D. So understand this very, very well, and we will have an analogy of the collagenase and trypsin EDTA with this as well. So let's take a look at what happens. So the plasminogen activator is essentially produced by the cell, and the function of the plasminogen activator is to activate proteolytic enzyme on the surface of the cell. So again, just to remember that what we are saying. So this is, you have the cell. On the plasma membrane, there are several proteolytic enzymes, and these have many functions that they process molecules around the plasma membrane on the external surfaces. And these are, again, we just naming them as a proteases. These are proteases or proteolytic enzymes, proteases. Now, these proteases remain fairly inactive. But what happens that when the cell actually gets a signal that these proteases should work, it produces a plasminogen activator. So how the plasminogen activator works, it will be synthesized through the secretory pathway, and the plasminogen activator through the Golgi will be loaded onto the vesicles, and this plasminogen activator will be targeted onto the plasma membrane, which will activate the proteases, Again, repeating, plasminogen activator produced, causing external proteolysis means by activating proteases, cell plasma membrane, or I'm just writing PM proteases. So just remember that this is how it works. So two things happening: production of plasminogen activator that will activate the plasma membrane proteases for increased proteolysis, means that the action of proteases are proteolysis or proteolytic digestion. So why this is important, just a little bit of off topic, that we all know that for your coronavirus, so for COVID-19, the ACE2 is one of the receptor for that. And you all know that there are some proteases that are usually involved in basically cleaving a portion of that spike protein of the COVID-19, so that the fusion peptide is essentially exposed and it helps in the entry. And this protease is which actually cleaves that spike protein of this virus is known as it's called TMPRSS, TMPRSS2, TMPRSS2, and this is essentially one of these proteases. There you may hear that there are some called furin-like proteases. These are some of the proteases which are all plasma membrane PM proteases. So this TMPRSS2, furin, these are present on the surface because they do a lot of different things and remodeling of different things, and they help in the normal physiology of the cell. But what happens that their activity, or the protease activity, is well regulated. They do not proteolyze too much because it could be detrimental to the cell. But what happens, the adherence abnormalities of how it is done, that when the cell produces this excess amount of plasminogen activator, that will activate these proteases more. Proteases activity, so I'm just writing protease activity on the plasma membrane will go up. And that is your increased extracellular proteolysis. Now, why it has to do with the adherence abnormalities? Think about what it is doing. So here is a single cell on this. And remember that adherence with the ECM protein are these integrins. And what is going to do that the proteases are going to partially digest these integrins and say the fibronectin, which are the ECM proteins. So all these anchoring proteins or the cell adhesion molecule or the CAMs are proteolized, because they are proteolized. So cell adhesion molecules are proteolized, and that's why. So when these are proteolized, the cell essentially floats, and that is your adherence abnormality. They do have the anchorage. They do not have the anchorage dependence anymore. Now, see that right here where you have the analogy. When we talked about the unit 1, the very beginning, the cells cultured, you have all the cells in here. And now when you passage the cells, you treat these cells with the collagenase, trypsin, and you also have EDTA along with that. If you just remember, take a look at those: trypsin, collagenase, EDTA, and they are used on these cells, and they help to the cells to move or detach from the bottom. And once they are detached from the bottom, you can just again take them and place them in a new Petri plate. Of course, you just remove that collagenase, trypsin, and the EDTA media. You just put them on the cell, just allow them to act for about just 5 minutes. Then you just aspirate those out, and the cells will come loose. So what the collagenase trypsin is doing there, the collagenase and the trypsin, they are also nothing but the proteases. These are the proteases that the collagenase can easily, you can imagine that they will digest the collagen, which is one of the extracellular proteins and ECM proteins. The trypsin is also another protease, digest a variety of protein. So this is the protease that digest those cellular adhesion molecule. So they partially digest the CAMs by the proteolysis. So what is happening here, that in the cells, which are the transformed culture cells, naturally, the cell is producing excess amount of proteases by itself, and that is mimicking this condition, that you add collagenase trypsin during the passage to the cells actually come out of the bottom. When the proteases are produced by this excess amount of plasminogen activator signaling, the same effect is achieved. The cells actually become rounded up and they do not attach to the bottom, so they lose their adherence capabilities. So just remember that. And we also talked about that what was the function of the EDTA along with that. Remember that now you know that your integrin, even the other ones as well, the integrin or the cadherins, remember that these cadherins, they all require the calcium, Ca2+. And the EDTA is going to mop up all these calcium, and integrin will fail to work, and the cadherins will fail to work. And these are all your adhesion molecules. Cadherin is cell to cell, integrin is cell to ECM, and they will essentially work. Remember that integrin is the protein that you see in the hemidesmosomes, as well as you see them in the focal contacts everywhere with the ECM. So they will essentially lose. So just know that what is the collagenase trypsin? These do the proteolysis of the cell during the passage, but here the plasminogen activator produce the proteases, which is the same thing that you externally use. This is why you have these adherence abnormalities. Then normally the cells will digest these ECM, the cellular adhesion molecule. So know this very well. I repeated it a couple of times because it's very important. So if we just ask the question, it can be formed like this, that if we say to remove the cells from a tissue culture flask, say EDTA and trypsin are used, and then, or say EDTA, collagenase, trypsin, but anything, EDTA, trypsin are used, which of the following neoplastic phenotype of cancer cells you may consider as analogous function of trypsin? So again, repeating, to remove the cells from a tissue culture flask, EDTA and trypsin are used, which of the following neoplastic phenotype, which means that property essentially, neoplasticity is like a neoplastic property of the cell, transform property of the cells, you may consider as analogous to the function of trypsin. So absence of actin stress fiber, nope, that's not the correct answer. That's not the analogy. Reduce external coat of fibronectin, nope, that is not the analogy. That happens, but it is not the analogy for the trypsin. The inhibition of production of plasminogen activator, no, that is not the function of that as well, because the plasminogen activator essentially is an activator of the protease. By itself, the plasminogen is not a plasminogen activator is by itself not a protease. So that's the important part. And a release of, say, excess calcium in extracellular matrix, nope, that is not correct. So what could be the correct answer? The correct answer could be that, or will be that the increased extracellular proteolysis by proteases. So so right here, increased extracellular, increased extracellular proteolysis by proteases. Remember that protease word is not there, but it is assumed that when it is mentioned as proteolysis, this is done by the proteases, which is present on the plasma membrane of these cells. They are, so I'm just going to draw in a different color. They are present on the plasma membrane of the cells, but they remain in an inactive form. But in the neoplastic cancer cells, these plasminogen activator overactivate them, and that causes the increased proteolysis of the external cell adhesion molecule, or the CAMs, which is analogous to the CAM proteolysis, that EDTA trypsin, which you use during the passage of the cells. So very important. All right. So after that, we are looking at the next slide. It is equally important, the growth and the division abnormalities. So the growth to an unusually high cell density, that means that they do not have any contact-mediated inhibitions, and that's why they can do this. Lowered requirement for growth factors. And we are going to see that why it is a lower requirement of growth factors, because we are going to go into the molecular mechanism of that. Because what happens, the growth factor binds to a certain receptor on the cell, and it tells the cell to go into the division. That is the function of the growth factor. And so some of these growth factors are as simple that you have already learned in your signal transduction, is a receptor tyrosine kinase. So you have a fibroblast growth factor that is present in the serum that you just produced. And when this FGF, fibroblast growth factor or nerve growth factor, depending upon the cell, it binds onto the receptor tyrosine kinase. The receptor tyrosine kinase will autophosphorylate, it will dimerize, and then it will transduce the signal. If you remember that it have the first protein, which is the GRB2, the GRUB2, that will recruit the sauce, that will activate the RAS, then it will activate RAF, then it's MEK, MAP kinase, then P90RSK, and then this P90RSK and the MAP kinase all together will essentially increase the cell division or growth by binding to the DNA, which will regulate the growth cycle. Now what happens, sometimes you're going to see that some of the proteins, like say RAS, we are going to see that the RAS, which is in the pathway of that receptor-mediated activation, is mutated, and it is constitutively active. That means the receptor, without the requirement of growth factor, is a signaling cell division. And that's why they lowered the requirement of the growth factors. Without the growth factor, the cell is already signaling the cell to divide uncontrollably. And this we're going to see in a second. So that's what we mean by the lowered requirement for growth factors. Then we'll see that less of the anchorage dependence can grow even without the attachment to the solid surface. And we saw that. Loss of contact-mediated inhibition, cells pile up on top of each other, forming foci. We already saw that. And they become immortal, can continue proliferating indefinitely, because all the cells actually do have, normal cells do have a limited number of division in them. And the cells will normally, the cells will undergo what is called a programmed cell death, and that is known as the apoptosis. So this is one of the word. This word is probably used, I will say probably 99% of the folks pronounce this word as an apoptosis. But if you just look at the linguistic, that how the origin of this word actually happened, the second P should be silent. So the apoptosis is the correct pronunciation, but you will see that 99% of the people will, even like all renowned scientists and everyone, most of them will say apoptosis, and which we always take it for granted, but the correct pronunciation is apoptosis and the P is a silent. So just, and this is basically the programmed cell death. What means that after a certain amount of division, this programmed cell death or the apoptosis will essentially kill the cell so that it doesn't divide. And why this program is there, that when the cell is undergoing 50 or 100 division, the cell understands that there is accumulation of mutations and it is not safe to keep the cell alive because the cell may spontaneously go into a new plastic form. So the apoptosis essentially is a mechanism to restrict that cell division after a certain point. But when that control of apoptosis is gone, the cells become immortal, proliferating indefinitely. So there is no programmed cell death of apoptosis. So that's basically the loss of apoptotic control, or the programmed cell death control also leads to that, and also can cause tumors when injected to susceptible animals. Now, this is where this word is the most important word right here. So just remember that if you just have some tumor cell, and if you normally inject that to a normal animal, it's not going to cause cancer because you already have an immune system that will find these cells and kill these cells. The susceptible animals are immunocompromised. So this will happen only to these type of animals, that if they're immunocompromised, doesn't have a deficiency in the T cells or the B cells, T lymphocyte or a B lymphocyte. Usually those are basically meant by the susceptible animals. But just remember that word is important. All right. So typical type of question that we can ask is, say, for an example, compared to a transformed cell, a normal cells in culture have enhanced rate of a cell division, form the foci, have lost contact mediated inhibition. So if we just give that, none of these are correct, because remember that, again, I'm repeating, compared to, so essentially just which of the following is the correct answer is the question. And if we say this statement, compared to the transformed cells, normal cells in culture have enhanced rate of cell division. Nope. Form foci like this. No, normal cells do not form foci. See that these cells are flat on the surface. They do not form foci. Have lost contact mediated inhibition. No, the normal cells have contact mediated inhibition. When they touch each other, they will stop growing. It is a part of the contact cells. But if we say that compared to the transformed cells, normal cells in culture, they are mortal, that will be a correct answer, because the transformed cells are become immortal, but the mortal cells will be mortal. But if we just have some of these, like say, have enhanced rate of cell division, form foci, have lost contact mediated inhibition, all choices are correct. None of the choices are correct. If we just have these choices, just make sure that if they're all wrong, feel free to choose. None of the choices are correct. It may be given all the properties of the transformed cells and none of the properties are of the normal cells. And in that cases, none of the statement could be correct. So just feel free that to choose none of the choices correct. So these type of questions that we can ask. All right. So after that, we are looking at a transfection assay. So what it shows that this is basically the experiment that tells you the last line of this, that caused tumor when injected to susceptible animals. And that essentially explain this. So what you can do, that you can isolate this tumor and then you can isolate the DNA from there. And then you take a mouse fibroblast and you transfect that tumor DNA into these fibroblasts. So this is one of the cell that these cells will essentially have a transformed cell or a foci. So if you take these transformed cells and inject to a normal mice, this will not have a tumor. But the mice that you are seeing, that there are two different types of mice that are immunocompromised, and these are the susceptible mice. They are either called SCID or nude mice. So the SCID is the severe combined immunodeficiency. So the SCID mice do not have T cells and B cell. They are T and B cell deficient. So none of the T and the B cell can be active. So the function of again, these are not needed. But if you remember that the T cell can be a cytotoxic T cell, which essentially find the tumor tissues and directly kill those tissues. Whereas the B cells essentially secrete the antibodies, which can bind to the abnormal proteins, and other immune cells can detect those and kill those cells, abnormal proteins. On the nude mice, on the other hand, is only T cell deficient. So you can see that these produce these cytotoxic T cells, which can have the ability to kill the cancer cells as well, which are the abnormal cell. So any of the two can be used, but the SCID is more immunocompromised than the nude mice. But these are the susceptible animals. So usually they do not have the skin, and in the laboratory, you have to maintain them in the animal facility in extremely almost sterile condition, because remember that as they do not have the T cell or the T or the B cell, they are susceptible to all the environmental pathogen, and they can die very, very easily. They are extremely susceptible and have to be kept in an extreme sterile environment, and has to be handled like this experimentally. All right, so that's all pretty much. It's what is meant by the susceptible animals. All right, so after that, we are looking at the proto-oncogene and the tumor suppressor gene, and the part 2 and 3 will be those. So these are some of the very important definitions, and just know this, what we mean by these things. So the proto-oncogenes play a role in normal cell growth and proliferation. And these are the positive regulator. Now, the name is a misnomer. They are not there for oncogene or tumor-causing genes. They are not. When they go out of control and malfunction, then they become oncogenes, which actually lead to uncontrolled cell growth. So, but these are not that these genes are cancer-causing genes. These are cell cycle control genes, and these are the positive regulator. And the mutated version of the proto-oncogene is a gain-of-function mutation contribute to transformation of the cell, and they are usually dominant. So let's dissect that, what we mean by this and what is the positive regulator. Just remember that the proto-oncogenes are always, normally they are off. Normally they are off all the time. And they are only when you get a signal. And this signal is essentially a growth factor, GF, growth factor. Comes from the cell culture or anything, or in the body, the growth factor. So when you have the signal of the growth factor, these normally off genes, they will be turned on. So when these genes are turned on, means that these genes are producing the product or a protein that will bind onto the DNA and will tell the cell to divide. So this is a positive regulator. When they are on, they will tell the cell to divide. Normally, they are off. Remember that. So what happens, the mutated version gains a function. So when the proto-oncogenes normally should be off, gain of function means that this capability goes off. It just becomes constitutively on some way, constitutively on or duplicated, or have hyperactivity, but constitutively on. And this will have unregulated cell division and control, and that's what the problem is. And that is why you can see that without the signal, which is the growth factor, if there is a mutation of these genes, without the growth factor, it will be constitutively on, and this basically becomes a proto-oncogene, becomes an oncogene at that time. So gain of function means that suppose the alleles are, say, for an example, just think about this way, that you have the three different conditions. You have the AA, and then you have an A, small a, and small a, and like this. So these are the three conditions: person 1, person 2, and person 3. Think about that your capital A is an oncogene. This is mutated. This is mutated. Whereas a small a is non-mutated. Non-mutated, which is a proto-oncogene. That's a normal. So see that if you have the normal gene right here, small a, which is non-mutated, that's a positive regulator. That means that it is constitutively off and it can be turned on only upon cell division. That's perfectly fine. But the capital A is mutated form. That means that it is constitutively on. And of course, if it is a homozygous, both the alleles, it is on. But here, in this one, where is heterozygous, this one is on, whereas this one is off. But this on is good enough, and that's why we call them as a dominant. So this person and this person, or these two different type of cell, they will lead to the cancer. And that's why we call this as a dominant. Whereas this one becomes the normal, this will not become normal. So just remember that the small a is the non-mutated form, capital A is the mutated form. So you can see in a heterozygous condition, it will also cause the cancer. That's why we call is a dominant. And again, take home message, they are constitutively off. Only upon signal, they should be turned on. But the gain of function is essentially they become constitutively on somehow. That's your proto-oncogenes. Now let's take a look at the tumor suppressor gene. This is the exact opposite. Normal function in inhibition of the growth and proliferation, and it's a negative regulator. So the tumor suppressor gene is on all the time. And a classic example of RB, if you remember that this is on all the time. And when this gene is turned off, when these genes are turned off, that will be the loss of function, and that will lead to cancer. So the difference between this is constitutively off all the time, when it becomes on constitutively, that can lead to the gain of function. This is on all the time, and they are only off. So just remember that the off does not mean that always it will cancer. So the off means that when they are off, when the cell gets a signal or a growth factor, only at that time make these genes temporarily off so the cells can divide. So they keep the cells checked so that they cannot divide. Only upon getting a signal, it will turn off. And then once the signal is gone, it will again go back and turn it on so the cells have a regulated cell cycle. But when they become constitutively off, and which is the loss of function, that they cannot be turned on, that can lead to the neoplastic cells. And these are essentially recessive. So let's see that what is happening in here. So here also, A is normal TSP. TSP means tumor suppressor protein or gene. TSP, TSG. The tumor suppressor gene produces the tumor suppressor protein. And AA is the mutated, and this is loss of function. And so you have your AA capital. This is normal. That means they are both on, no problem. Here you have A and small a, and see that this is on, this one is off. In both the cases right here, you do not have problem. Just one of the gene is functioning and producing the tumor suppressor protein, the tumor suppressor gene, and that will check the cell cycle. But if you have the AA like this one is both the alleles are mutated, and in this case, these are the ones where both of them are constitutively becomes off due to a mutation, and that can lead to the cancer. So here you can see that's why we call that they are recessive. If they are in a heterozygous condition, the tumor suppressor gene in heterozygous condition will not cause tumor. Only in the homozygous condition where both the alleles are defective, it will cause. But if you just compare that to your proto-oncogenes, remember that the proto-oncogenes produced cancer in this case and this case. Both cases, it produces the cancer. The only case that did not produce the cancer is that here, that is a normal, where this was the normal. Whereas it's just the opposite for the tumor suppressor gene. The homozygous normal as well as the homozygous heterozygous, one normal, one abnormal, they do not have the cancer, and only in the homozygous mutated form they will have the cancer. So just the contrast in between them, that why this is dominant and why this is recessive. Just remember that they are always constitutively off, just only turned on, and these are always on and only turned off due to a growth factor. So the typical question that we usually ask out of that, say, which of the following statement best describes the relationship between the tumor suppressor proteins, TSPs, and the cancer? Again, remember that tumor suppressor protein is the protein produced by the tumor suppressor gene. So if we just make a statement like that and say that, let's discuss some of the distractors, which are incorrect answer, that which of the following statement best describe the relationship of between tumor suppressor protein and cancer? So if we just have a distractor like this, which will be incorrect, protein encoded by oncogenes will bind and activate TSPs. No, that is not correct. Proteins encoded by the oncogenes will bind and activate the tumor suppressor proteins. No, that is not correct. They are entirely different things. When activated, the TSPs will cause uncontrolled cell division. No, that is not correct. Remember that when activated, TSPs will cause uncontrolled cell division. No, when activated, their function is to suppress tumor. So when they are activated, they are controlling cell division, not. So I'm saying that when activated, TSP will cause uncontrolled cell division. That will be just the opposite statement. So that will be incorrect. When activated, TSPs will block the activity of, say, P53. So P53, we're going to see that in a second. That will make much more sense. Protein, which is we call is a guardian of the genome, that will block the cell cycle essentially. It will not block the activity of the P53. We're going to see that some of these tumor suppressor gene will promote or help the activity of the P53, not block. So what could be the correct answer? If we make this like this, the mutation to a TSP gene will cause a loss of function. Remember that. If it's a loss of function in the protein that it encodes, that will be the correct. And it's inhibition of the cell growth, proliferation. So it is not, so it is always on. Whereas these genes proto-oncogenes are only turned on during a cell division by a growth factor. And here the growth factor actually temporarily turn this one off so the cells can divide, and then automatically it will turn back on again so the cells don't divide. So spend a good amount of time, because this is a concept that is important in the next part as well. And a lot of people actually miss those questions. So that's why I just spend a good amount of time on this one. All right. So then we'll go quickly through here. So here, what we are looking at, that the induction of cancer requires more than mutations. And you can see that we have like a human being have a 10 to the power 16 cell division, 10 to the power 6 mutations per gene, per allele, per divisions. So 10 to the power 10 mutations of the genes in a human lifetime. But compared to that, when we look at this graph of these per 100,000 people and the age group, this is the occurrence or the percentage of the people that you have the cancer. And you can see that as we age, or a population actually ages, the cancer actually goes up. And why this is so? Because this is the most important idea. You need at least 3 to 7, minimum 3, as many as 7 independent genetic accidents required in the same cell. So the thing is that in our bodily cells, these mutations, 10 to the power 10 mutations in the lifetime, mutations are happening randomly all the time, but they are happening in different genes, different cells. So some of the cell cycle control protein can be mutated, but when a cell cycle control protein 1 is mutated, there are other proteins which can control that mutated protein as well in that cell. If there are other proteins which are looking after the cell cycle control, all of them which are the guards, those are also mutated, then it may lead to the cancer. So just only one gene will not cause that. And what happens, the chances of accumulating those mutations grow over the period of time. So why this graph is like this, just remember that the age is directly proportional to have these genetic accident or mutation and increased probability of cancer as we age. Because see that what is happening here, just an idea, that suppose a person has one mutation here and one mutation happened right here. And then a person have another mutation, the second mutation happened in here. And then a third mutation happened in here, which may lead to the cancer, that 3 to 7. On the other hand, a person that they are having random mutations, but they are in certain genes that this is 4, 5, 6, 7. Until all of these genes are mutated, that person will not show a cancer. And it has to have in the same cell. Remember that you have trillion cells in your body, and it is a single nucleus. So that these cells divide into this and then divide into this. And that's what we say, that a cell divides about 50 to 100 times. And after that 50 to 100 times, the cell is killed by apoptosis or the programmed cell death, so that these mutations do not accumulate. But the chances of the mutations to accumulate grows over the period of time. And that's where the increase. And the reason is that what it shows, these graphs, that why the cancer occurrence grows exponentially directly proportionally with the age group, is that multiple mutation is required for that. So we can give you a graph and then say the incidence of colon cancer increases with age, as shown in the accompanying figure. Like we will give you a figure like this, where the number of newly diagnosed cases in women in 1 year is plotted against the age of the diagnosis. So just think about that. We just give you a graph like this. And here it is that cases in women in 1 year plotted as a function of age in the diagnosis. So this is increasing age. Remember that. So like this. And when we see that how would you explain these data? The data explanation is the graph pattern will be the same. And essentially the explanation is, again, it takes mutation in several critical genes to disable the cell's normal mechanism of controlling its growth. So that's the point. It takes several mutations, several critical genes to disable the cell's normal mechanism to control growth. Just one protein goes wrong, there are other proteins which can actually help the cell still to survive without losing the control. But those critical couple of them, and that's why that with age, it goes down. So these are some of the things that is important. Again, remember that multiple things are important. So on the other hand, the distractor we can use, say, for example, women are more susceptible to colon cancer than men. It's actually the opposite. So that is incorrect. Colon cancer are more difficult to diagnose. The diagnosis has nothing to do with the frequency of the cancer. Colon cancer caused by novel fusion proteins, yeah, that is correct. We're going to see that. But that has nothing to do with the occurrence of that. That is a mechanism of colon cancer. The occurrence of colon cancer with age increase doesn't corroborate with that. And colon cancer treatment is less effective in women. In men, that these also doesn't have anything to do with the occurrence of cancer. So the answer that is the most rational is that it takes mutations in several critical genes to disable the cell's normal mechanism of control. So another type of question we can ask is that efficient oncogenic transformation of the rodent cell. So we already talked about this on the very beginning. So if you actually have normal human cells, over the period of time, they will grow primary cells and then they will die. You saw a graph like this that when in a very first few slides of your Unit 1, Module 1. Whereas we show you a graph of the rodent cell that they actually die gradually, and then sometimes after the senescence, this is the area of senescence, they can actually go back and become spontaneously transformed. And why this happens, that this requires mutation of at least 3 or more of the oncogenes so that it can essentially happen. So that's the important one. So that the efficient oncogenic transformation of rodent cell requires a mutation of at least 3 or more proto-oncogenes. And we are going to see a question based on that. So here we are looking at the tumor appearance in mice overexpressing these MYC and the RAS oncogenes. The scientists actually developed some of the transgenic mice where they have these MYC, which is a cell cycle control, RAS, but this is your RAS with valine 12, this is the mutation. Then this is the RTK pathway protein, the signal transducer that we talked about. So these two proteins were mutated, and then they actually transgenically put these into the mammary tissues of these, where they have these mutated cells by the transgenic technique. And what they did, they put these genes under a viral promoter, which is called a mammary tumor virus, MMTV promoter. And here you have either the MYC under that, or you have the RAS. And what happens, the interesting thing is that MMTV, this is the promoter. That means that this will drive the expression of these mutated MYC or RAS in the breast tissue. And you can induce this by the hormone. So you can just feed the mice some estradiol or estrogen, the hormone treatment, and this MMTV promoter will be turned on, and these proto-oncogenes or the MYC or the RAS will be turned on. So when you turn them on, and that will essentially lead to the cancer. What it shows that if you have only MYC, here the mouse have only MYC, say that you are producing about 2 months it takes the tumor to form. RAS is much more aggressive. You can see that just within probably about 30, 40 days, you will have appearance of the tumor. And say about if you can see that even after about say 100 or 200, almost 200 days, so you can easily see right here that probably this is the percentage of the mice that is tumor free. So even after about 3 months, almost 80% of the mouse is tumor free. Whereas the RAS, you can see that almost like 50% of the mouse are tumor free after 3 months. But you can see that within 150 days, if you have RAS and MYC together, just 150 day, you have no mice that is essentially surviving. And this is basically what we call as the synergistic effect, a synergism of the tumor proto-oncogenes and that's basically what it is. That when we say that the efficient oncogenic transformation of rodent cell requires mutation of at least 3. So when you just have, so this is in the mouse model means this is essentially a mutated gene in the mouse. This is a particular experiment. Remember that this is not normally even happen. It is under a specific promoter that you are turning on and all those kind of things. And it just shows the synergism. But if the MYC and RAS is just normally mutated in some of those cells, there will be needed additional mutation because they will not be always turned on under the normal promoter. So just remember that promoter that underneath that, just that RAS or MYC mutated does not mean that this promoter is activated all the time. So this could be, there should be a problem in here as well. So you have 1 + 2 + 3 or even more, because sometimes these promoters have multiple things. So multiple thing has to go wrong. So it needs at least 3 or more. So just to remember that to transform. So that is your number. So your magic number is essentially that 3. That is the minimum number. Remember that. All right. So let's go into this one. So after that, we are looking at last slide of the general consideration. So we have the proteins that function in cell division and control. So the proto-oncogenes are class 1 through 4, and the tumor suppressor gene are 5, 6, 7. So the first one is class 1 is the different soluble signaling molecule. And these are the soluble hormones and all those kind of things. Sometimes they can mutate it as well, and they can aberrantly bind to different things. We're not going to discuss that too much. So, but they are there, so they can be mutated form. Then we have a signal receptor, which is a class 2 we are going to talk about. These are intracellular effector region, often a protein tyrosine kinase. We are going to see that. This is your RTK. We're going to see that. Then we have the intracellular transducers, just like RAS. We are going to see that. We're going to talk about that. So signal receptor, intracellular transducers, we talked about. And then you have the second messenger. You can see transcription factors. So here you have the intracellular receptor. That is also the same way as the signal receptor. then your transcription factors are the fourth type of the proto-oncogenes, as you can see. That usually the transcription factor, remember that. Why proto-oncogene? Because you see that all of these proto-oncogenes, 1, 2, 3, 4, if you carefully look at that, all these genes are normally off. When they are on or gain of function unconstitutively, and at that time, only they become the cancerous uncontrollably. Remember that. They are supposed to only turn on due to a proper growth signal, but if they're not. On the other hand, if you see that the class 5, 6, and 7, so 5 cell cycle control protein, then 6 DNA repair protein. Now, the 6 is not exactly a tumor suppressor gene, but we classify them as a tumor suppressor gene because see that DNA repair proteins essentially repair a damaged or mutated DNA. So if DNA is mutated, where these DNA damages, if they are within a tumor suppressor gene, that is making the tumor suppressor gene constitutively inactive due to the mutation. If the DNA repair protein doesn't repair that, then the tumor suppressor gene function is gone. So it will not be able to have the tumor suppressor function. That's why they are also under the tumor's DNA repair proteins, but they are not directly tumor suppressor genes. On the other hand, we have seen that apoptotic proteins, the class 7, they are also tumor suppressor genes. We are going to see that, say, for example, P53 is one of the function of that is apoptosis, which is essentially if the cell undergoes uncontrolled cell division, the cell has the ability to detect that and kill that. If that protein, apoptotic protein function of uncontrolled cell growth or the tumor suppressor function is gone, then the cell will uncontrollably grow. So these are the different classes of those. And just to remember that what are the different types. All right, so let's go on to the next part. All right, so in the part 2, what we are going to see that in the part 2, we are going to talk about the proto-oncogene, and the part 3, we're going to talk about the tumor suppressor gene. So the part 2, we are talking about the proto-oncogene. Now, remember that we said it already several times established that the proto-oncogenes is a dominant gene and it is a gain of function mutations. And the proto-oncogenes are constitutively, that's again, I'm just repeating all the time, that they are constitutively off. They are constitutively off and they are on only upon a growth factor or signal. So what are the different gain of function that can happen? So here, there is a deletion or point mutation in the coding sequence. So you have the DNA and a sequence, there's a mutation. And here, constitutively on, or it becomes a hyperactive protein. Whereas here, what is happening is the protein is the gene, there is no mutation in the gene, but the gene actually amplified. So there is one copy, this is 1, this is 2, this is 3. So one copy became 3 copy due to the duplication of the genes. So it's going to produce 3 times the normal protein. So the normal protein is overexpressed. So when the protein is overexpressed, that will overstimulate the cell cycle growth. So here, there is no mutation, but just the duplication of the gene or gene amplification causes the proto-oncogene a gain of function. Here, chromosome rearrangement. So here what happens that we just did that experimentally for the MYC and the RAS in that mammary tumor virus promoter with the mammary tumors virus promoter, that the experiment we talked about. Sometimes in the chromosome rearrangement, proto-oncogene can insert itself under a strong promoter and say, for example, we say that that promoter is constitutively on. So what it means that this is the switch. So in a normal proto-oncogene, why it is constitutively off? Because the promoter that it has, which is the switch, is off normally. But if the gene actually cut itself out, and these movements can actually happen in the chromosome due to the transposon and transposition constantly happening, and if they by chance move into a region where you have a promoter right here, and this promoter is a strong and it's constitutively on instead of off, it is going to drive the expression of the gene always on. So the gene becomes constitutively on as well. So normal protein overexpressed, and you can see that the function is the same, that it will drive cell cycle into a faster pace. On the other hand, you can see that here you have a chromosome rearrangement, and here you have a fusion protein. There are two differences in here. Remember that a promoter region is upstream of the DNA, where it binds to a region where RNA polymerase bind to make these mRNAs. So this is the promoter is not a part of the gene. It is a control or regulatory area of the gene, which is it is expressing. Here, there is a different protein is fused essentially. So the gene, this is a gene fusion. This is a gene fusion, just randomly, a random gene of an entirely different unrelated protein can join, and the protein is truncated, both of them truncated and just join, and it's producing a fusion protein. And sometimes this can produce a constitutively on or hyperactive protein. And how this happens, we are going to see. So in all of these cases, this can lead to the constitutively on condition or overexpression of the gene, hyperactive gene, and that can lead to the cancer. So the proto-oncogene can lead to the oncogene. So just remember that if we just look at the mutations, where the mutations are happening, here there is a mutation, here this is a mutation. So remember that this is a mutation and there is a mutation. That means that like really the sequence of the original gene is compromised. And here there is no mutation. Just remember that here is no mutation, here is no mutation. But what happened, that's due to the amplification of the gene, which is normal, the normal protein is overexpressed. Here, there is no mutation in the gene, but due to a stronger promoter, normal protein is expressed. But here, it's a hyperactive protein. It's mutated, hyperactive fusion protein, or constitutively on, and these are the mutations. So just remember that which cases you have these mutations, which cases you do not have the mutation, and how you gain these functions. We are starting with the class 2. We are not talking about the signaling molecule. These signaling molecule could be a wide array of molecule which can become aberrant, and then they can bind and start a signaling process that can happen. But the class 2 which relate to what you already know. So these are the receptor tyrosine kinases, or the RTK, that we talked about in the signal transduction. And see that how this activation actually happens. This is a known material, remember that. So you have the monomeric receptor tyrosine kinase. Upon a ligand binding, the ligand is, remember that this ligand is nothing but a growth factor. These ligands are the growth factor. When it binds, the RTK dimerizes, and then it's cross autophosphorylates. Remember that it cross autophosphorylates other tyrosine residues. That's why we call this a receptor tyrosine kinase. And then you have the other proteins that we did not talk about. You have the adapter proteins. So remember that you already talked about. You have the GRB2. Then you have SOS, if you remember from your previous chapter, which will activate the RAS. So the RAS is the first protein, which is the transducer. So this is your transducer protein. So basically what it is doing, that this is the protein which is transducing the signal received by the receptor into the cell. GRB2, this is an adapter protein, and the SOS is essentially with the help of GRB2 binds to the RTK, and SOS is a guanine nucleotide exchange factor. Remember that the RAS acts with it is bound with a GDP in an inactive form, and it is replaced by an GTP in the active form. And the SOS actually does that replacement of the GDP to GTP to make it active. And then it activates the RAF, MAP kinase, and transcription factor, and its growth. And this protein can go wrong. And let's take a look at what are the things that can happen. So here is a chimeric oncoprotein from chromosomal translocation. Where is an example from the first slide? So this example of the receptor tyrosine kinase is this example. Remember that. It is a fusion protein and it's constitutively on fusion protein. So this is essentially the example of that receptor tyrosine kinase in that cases, and this is implicated in colon cancer. So let's take a look at these two entirely unrelated proteins. One of them is a RTK. Sometimes it's known as a tyrosine kinase, TRK. If you just see that, they're the same thing, receptor tyrosine kinase. So this is a monomer of receptor tyrosine kinase, two monomers you are seeing, normal tyrosine kinase receptor. So here you have the extracellular domain. You can see that. Then you have a transmembrane domain. And then this is the C-terminal, which is in the cytosol. And you have the kinase domain, which cross-autophosphorylate upon binding of essentially the binding of the thing. So this is the N-terminal of the protein. This is the C-terminal of the protein. So just make sure that you carefully see that this is a transmembrane protein. This will be synthesized through the secretory pathway. That means that protein will be inserted into the ER membrane. From ER membrane, it will be packaged into the vesicle, will go to the Golgi. From Golgi, it will be loaded onto the vesicle membrane. That vesicle will go and fuse to the plasma membrane of the cell, where it will be delivered onto the plasma membrane. That is the pathway that RTK will be synthesized. So that's number one. And remember that N-terminal is on the external side. So when you have the N-terminal in this way, when it is in the ER, N-terminal will be the lumen of the ER, the N-terminal will be the lumen of the Golgi, the N-terminal will be the lumen of the vesicle, and then it will be flipped outside when the vesicle fuses with that. This is from your previous understanding. And then you also have this is a normal non-muscle tropomyosin. And it is a dimer, so N-terminal on this side, C-terminal, and this is a soluble protein. This is present in the cytosol. This is not a membrane protein. So it does not have any address or signal, because remember that if a protein doesn't have a signal or anything, it ends up in the cytosol. So this is an example of that. On the other hand, the RTK, you have an N-terminal, you have a signal that is inserted into the membrane. So these will have the signal to go and synthesize through the secretory pathway. Now what happens at the gene level, that this tropomyosin is fused with the RTK. So the way it is fused, usually due to the chromosomal translocation, if you just look at the length of that, it is actually made that, see that this length is shorter and this length is shorter. So essentially what happened, a part of the gene from here is cut out right here. So this is truncated here, and part of the gene is cut out here. It is truncated here, and it is fused in here. So this is exactly what happened. So where the truncation happens, so remember that this is your N-terminal, this is your C-terminal. This is your N-terminal, this is your C-terminal of the protein. So this is in the gene, but this is the sequence. So the C-terminal of this tropomyosin is truncated, whereas the N-terminal truncated. So when the protein is synthesized, let's see that where the truncations are. So when the truncations happen, tropomyosin, the truncation happens at the C-terminal. Suppose this part of the protein is gone. Whereas for the receptor tyrosine kinase, the truncation happened just above this transmembrane domain. See that this is the transmembrane domain. Remember that this is a dimeric RTK sideways. This is straight onto the plasma membrane. Here it is sideways. So this part of the RTK is gone. It's not there anymore. And this is the N-terminal. And see that what is happening here. The N-terminal contain the sequences for directing it to the ER. That is gone. It's not there anymore. And here the N-terminal doesn't have any signal, and the protein is now synthesized as a cytosolic protein. It doesn't go onto the ER. It doesn't go onto the membrane. It remains in the cytosol. And what happens? Without even the ligands need to bind in here for the dimerization. The protein dimerizes automatically in the cytosol. This is the fusion protein in the cytosol autodimerized. And what it means that this protein is constitutively on all the time, that means that this will bind to the GRB. This will bind to the GRB2, SOS, then RAS, then RAF, MEK, MAP kinase, cellular responses, and essentially uncontrolled cell growth will lead to the colon cancer. So you can see that a receptor now in this condition, this cell will not require growth factors. So we were talking about the cells have diminished growth factor requirement in the cancer cell. Now you know how. Because what happens that receptor tyrosine kinase, when it is on the plasma membrane, the whole pathway is essentially off. Only when the growth factor binds to them, it dimerizes and start the pathway to turn it on the cell cycle. Here, the truncated protein is constitutively on hyperactive and will not require any growth factor without any signal. It is constantly on, and that will lead to the cancer. So the N-terminal of extracellular domain of RTK is truncated and fused with the C-terminal truncated non-terminal myosin. So again, remember that where this is happening is important. See that here, this N-terminal is deleted, that N-terminal is not there. And for the tropomyosin, this C-terminal is deleted. And this is basically the orientation sideways. So you are looking at the protein sideways. See that this was in the membrane, transmembrane domain. And the bottom part, which is the activation domain, remains in there, but the activation for the ligand binding domain is not there. But this activation domain dimerizes automatically as they are synthesized. So we can ask the question, is there, which of the following fusion protein is often implicated in the colon cancer? Remember that a non-muscle tropomyosin fused to the N-terminus of a receptor tyrosine kinase is completely cytosolic. This is the most important one, is cytosolic in here. So that cytosolic word, definitely put that one in there. So what could be the other distractors, say, which are incorrect? So say, for example, if I say a non-muscle tropomyosin fused to the N-terminus of a receptor tyrosine kinase that is inserted to the outer leaf of the plasma membrane, that would be incorrect. It is not on the plasma membrane anymore. So that's why it's incorrect. A non-muscle tropomyosin fused to the C-terminal of the receptor tyrosine kinase and put on the slit of the plasma membrane. First of all, it is not fused to the C-terminal. It is fused to the truncated N-terminus. And second of all, it is not going to the membrane. If anything says that chimeric protein is going to the membrane, that will be essentially a wrong answer. On the other hand, if we say a non-muscle tropomyosin fused to the C-terminal of the tyrosine kinase, that is completely cytosolic. So what is the take-home message from here? Normal tropomyosin that fused to which terminal? N-terminal of this protein. So that is what you definitely need to remember. Plus, they are cytosolic. So this entire protein is a cytosolic, not on membrane, not on membrane. So these two things are very important, that where it is going at the N-terminal. So if you just remember this picture, it is not at the C-terminal. That will be incorrect. It's at the N-terminal, and it is not on the membrane. It is cytosolic. All right. So after that, we are looking at, say, for example, this is a HER2 receptor and an EGF receptor. These are essentially receptor tyrosine kinase. These are specific names. Remember that receptor tyrosine kinase is just a class of receptors. And here, just a single mutation, a valin mutation, can lead to the cancer. Just one mutation in there that can make these protein constitutively active means that they can dimerize without the binding of the protein. So these valins, when they are valine is replaced by the glutamine right here. So here they attract each other and dimerize. And we call this as a new oncoprotein. And in breast cancer, this is one of the ones implicated. On the other hand, this is an EGF receptor and epidermal growth factor receptor. And sometimes the N-terminal is deleted. And still, it does have the membrane insertion signal. It still goes to the membrane because you have a hydrophobic signal in here. And this is known as an ERB-B oncoprotein. And this is a constitutively active protein kinase. But this is, again, this is a membrane truncated oncoprotein. This is not soluble protein, just the fusion protein that we saw. And this is also implicated in many cancers. So what type of question that we can ask? Just make sure that note this down very carefully, because we can use some of the words which prereq may have, but it may be new that we are introduced in here in terms of mutation. Say, for example, which of the following type of mutation can't give rise? So we are saying that can't cause cancer. That's basically we are saying can cause cancer. The question will tell you some of the examples that you see here and then the previous slide. Those will cause cancer, and then there will be the correct answer, which is the one which will not cause cancer. So which of the following can't cause cancer? That's basically asking. Which of the following mutations cannot cause cancer? Mutations cannot cause cancer. Say, for example, let's see that which can cause cancer first. So which will be the distractors? So a point mutation changing just one amino acid in the protein product. See that from valine, you change to glutamine. So just one amino acid, one AA. Yes, this can cause cancer. So that is not the answer. On the other hand, say, for example, a translocation which puts the gene under control of a strong promoter, producing overexpression. Let's go, and because this question will have multiple example. So here, the second choice could be like this, where you have a translocation which puts the gene under a control of a strong promoter and producing overexpression. This can also lead to cancer. A point mutation which produces a stop codon, prematurely terminating, producing a truncated protein. So what can happen that sometimes you can have a truncated protein, and sometimes this truncated protein can become constitutively active, and that can lead to cancer as well. On the other hand, what is the important one that makes sure that you have, so again, truncated proteins can also become hyperactive and constitutively active. But if we give you an answer choice like this, a translocation that result formation of a novel fusion gene and a fusion protein product. So what we say is that a novel fusion protein and gene product. So this is the example, just the previous one. See right here. So this is basically a novel fusion protein and a fusion product. So we gave you, so when we are saying that which of the following mutation can't give rise to oncogene, we give you an example of this, a novel fusion protein, a fusion protein gene oncogene. We give you an example of, say, for example, translocated gene under control of a hyperactive promoter and a normal protein overexpressed. Then we give you example of one amino acid mutations, truncations, or a premature stop codon. Remember that is the ones that is not there, but just remember that that is important essentially. So just remember that truncated protein, stop codon. This can lead to hyperactive protein as well. That can lead to cancer as well. But if we have an answer which can't cause cancer, that if we see that addition or deletion of base producing a nonsense message and an inactive protein product. So what is a nonsense message? See, for example, when you have these different mutations, you have many different things. Say missense means that there is a mutation that will produce a different amino acid or something like that. Nonsense mutations are usually, it will produce codons which will not essentially make any sense, and it will not give rise to a protein that is not active. So the nonsense mutation produces an inactive protein. So as soon as you see that, this is the trick, that as soon as you see that the word, even if you do not remember that what a nonsense mutation is, An inactive protein can never give rise to the cancer. That is the only thing you have to remember. Remember that everywhere these oncogenes are becoming hyperactive, overproduced, those are the things active. But they are, if we say that it's an inactive protein, that cannot give rise to that. So again, repeating that which of the following types of mutation cannot give rise to the oncogene, your correct answer is any addition or deletion of a base producing a nonsense message and an inactive protein product cannot give rise to the oncogene. Which of the following can give rise to an oncogene? Again, repeating, which of the following can? So the other way, a point mutation that is changing just one amino acid, a trunk translocation that puts the gene under the strong promoter, producing overexpression, we saw that. A point mutation that produces a stop codon and the message producing a truncated protein, you saw that. And then you have a translocation that results in the formation of a novel fusion gene and a fusion protein. We saw that just in the previous slide. That's the example of that. All of them can lead to the cancer. This is a question that is missed by a lot of people. So just remember that what we talked about, nonsense mutation and an inactive protein cannot lead to a cancer because that protein will essentially be destroyed by the cell. All right. So after that, we are looking at the class 3 intracellular signal transducers, which is the RAS. And again, remember that we are just talking about the RAS is essentially the GF or GNRP, which is essentially the sauce, changes the GTP to GTP, which is active. And then to inactivate it, the gap or the GTPase activating protein enhances the GTP hydrolysis and it becomes inactive. And what happened, a glycine to valine mutation at amino acid 12, that's what we saw right here. See that RAS V12, that is the same mutation that we are talking about. A glycine at the 12th position is changed to V that can make this protein constitutively active. Or say Q67L, amino acid 67, that can lead to constitutively active protein. And glutamic acid to a leucine can change at 67 position, can do that. So this is intracellular signal transducer, which is the RAS. Remember that? The RTK pathway, that RAS that we are talking about. This is just only for informational purpose. What we can ask here is what way does RAS oncogene contribute to the cancer? That RAS essentially codes for a GTPase, which is mutated form, cannot be switched off. Again, repeating, the RAS codes for a GTPase. Remember that this is a GTPase protein and it cannot be turned off. So what could be the distractors? Say, for example, if we just write the RAS codes for a tumor suppressor protein, which produce abnormally large amount. Remember that this is not a tumor suppressor protein. This is a proto-oncogene, which is constitutively off. And when RTK activates it, only at that time it becomes active. So this is a proto-oncogene and becomes an oncogene in cancer. This is not a tumor suppressor gene. So that would be wrong. RAS encodes for a transcription factor, which produces an abnormally large amount. Nope, this is a signal transducer. This is not a transcription factor. Remember that the last things in the pathway of the receptor tyrosine kinase pathway, that the MAP kinase and then the P90RSK essentially phosphorylate the TCF and the SRF. Those are the, if you remember from those TCF and SRF, which bind onto the DNA, those are the transcription factors. The transcription factors are phosphorylated as the end product of the pathway, but this is not a transcription factor. It's a signal transducer. It transduces the signal from the receptor tyrosine kinase into the cascade of cellular pathway. Then we say that RAS codes for a truncated form of a growth factor receptor, which is constitutively active. Nope, this is not a growth factor receptor. Growth factor receptor is a receptor tyrosine kinase, and RAS is right here. So that's not correct. And if we say that a RAS codes for an unusual fusion protein that behaves like a constitutively activated growth factor receptor, nope. That is essentially, we are talking about a constitutively active growth factor receptor. We're talking about an RTK, but not RAS. So the correct answer for that is RAS codes for a GTPase. And in a mutation, it cannot be turned off or switched off. So this is essentially the take-home message. So where you have this, right here, intracellular signal transduction, this mutated RAS right here, the pathway starts with the receptor tyrosine kinase, which essentially can be mutated and can cause cancer. This one is a protein in the pathway. And the same way, just to remember that, we just did not talk about that the RAF-MEK-MAP kinase, any of these proteins in the signaling pathway are potential proto-oncogenes. And if any of these protein in this signaling pathway become constitutively on, they can keep the pathway on. And here we are looking at oncogenic mutation cause RAS to be constitutively on, keeping the pathway on, which leads to the transcription factor phosphorylation and growth and uncontrolled cell division. All right, so after that, let's take a look at the last one. So this is the transcription factor, and this is your class 4. So here what we have, that we are looking at two examples of two chromosomes, a chromosome 8 and a chromosome 14. And here there is a phenomenon which we call as a balance translocation that happens. Right here at the bottom part of the chromosome 8, there is this MYC gene, and the MYC gene that regulates the cell cycle growth, and it is almost all the time it is off. Remember that the off is the characteristic. So the promoter under which the MYC is, it is constitutively off. Only under the signal it will turn on so that the cell can divide. On the other hand, the chromosome 14 actually contain the areas of the genes which code for the constant heavy chain and the variable heavy chain of the antibodies. And this is seen in the Burkitt's lymphoma, which is essentially the B cell lymphoma. So the B cells, which is the B lymphocytes, grow uncontrollably, which leads to a leukemia. So it's a kind of a blood cancer. It's a B lymphocyte lymphoma, leukemia, blood cancer, whatever you say, uncontrolled B cell growth. So what is happening here? So if you look at the antibody, see that right here, the antibody has a stalk. These are the constant heavy region. So these are the constant heavy region. And whereas the variable heavy region, so the variable heavy region are those yellows right here. And the variable lights are the ones in the green. But these genes are the ones which are the variable heavy at the top in the Y-shaped part, in the prong, and in the stalk, you have the constant heavy. Now, remember that these genes are in the B cell, and the B lymphocyte constantly producing antibodies. So these genes, CH and the VH, are under a strong promoter, and they are constantly producing this because the B cell are producing the antibodies. So if we just think about that here the promoter is constitutively on, and here the promoter, the MYC promoter right here, that is constitutively off. So that's what's going on. Now, in the Burkitt's lymphoma, what happens that there is a balanced translocation of the chromosome, unfortunately. So this part of the chromosome breaks this part and is translocated on the chromosome 14. So this is a double-stranded DNA break and joining. So from here, the constant heavy and the variable heavy region, that actually is a variable heavy region that actually comes in here. Whereas the MYC, where MYC was, that MYC goes under the promoter, which is controlled by the constitutively on promoter right here. So now what happens, MYC is now constitutively on, and that will lead to the uncontrolled B cell division. And that happens due to constitutively on hyperactive promoter. So see that this is the word that how it goes. Translocation acquired immunoglobulin gene controlled region has constitutively active promoter enhancer. The gene is always on, driving transcription and resulting uncontrolled cell division. Because you have to produce the antibodies, it was constantly under the on, and the MYC from the off promoter now translocated onto the control of an on promoter, and that leads to this. So just remember that. So what type of question that we can ask from here? We can ask that which of the following is a characteristic mutational cause for aberrant MYC activity in Burkitt's lymphoma. Again, which of the following is a characteristic of mutational cause for aberrant MYC activity in Burkitt's lymphoma? Say, for example, the translocation, if we say that this will be the correct answer response, the translocation of the MYC oncogene under a constitutively active promoter enhancer region. So you can see that the one in the green, that is essentially the word by word your answer choice. But if we just see that what could be the type of distractors, we can ask is like an amplification of the MYC oncogene. No, there was no amplification of the MYC oncogene. It's a balanced translocation under a promoter. So there is no amplification. Fusion of the MYC gene to a highly stable nuclear protein. No, it didn't fuse with any nuclear protein or anything like that. Or deletion of the sequence that is usually destabilize the MYC mRNA. If it's destabilize any MYC mRNA, the MYC product, if it doesn't have anything with the product, then it will not drive cell division. So the product is correct. So there is no destabilization of mRNA or anything like that. That's not the correct answer. But the correct answer that the translocation is under a constitutively on-active promoter enhancer region that essentially is implicated in the Burkitt's lymphoma. We're starting with part 3 of the cancer cell biology, and this will be, so we talked about the proto-oncogene, and now we are coming to the tumor suppressor gene. And you already know some of the part of this, that RB is the first tumor suppressor gene that was identified, and function is the normal inhibition of the cell growth and proliferation. So essentially, these genes are constitutively on. And when by mutation or anything like this, these are turned off, they can lead to the cancer. So they are constitutively on. And when there is a signal for the cells to divide, they are temporarily turned off and then they turn back on. But mutation in the tumor suppressor gene can lead to the uncontrolled cell division. Loss of function mutation contribute to the transformation. Now, RB is a recessive gene, and one defective copy does not cause the tumor, but it is inherited in a dominant manner, which means both copies of RB are defective to show the dominance. That is expression of the tumor disease or the phenotype. So these two lines, a lot of people actually get confused with that. So just make sure that you understand that what it is, and we'll discuss that typical type of question that is asked. So RB is essentially a recessive gene. So let's draw a couple of these. So let's draw that this is A plus. Say, for an example, this is a normal gene, normal RB. And then A minus is defective RB. Defective RB. So what is happening in here? Just think about that a person has, these are the possibilities that you can have. You have an AA. I'm just first, all the possibilities. I'm just drawing all the possibilities. So you have A plus and A plus, A plus and A minus, and A minus and the A minus. These are the three possibilities that can have. You have an A plus, normal, homozygous. This is normal, abnormal, or defective, heterozygous. And this is abnormal or defective, homozygous. So first, the RB is a recessive gene. Why it is so? Because remember that we said that it is inherited in a dominant manner. That when you have the tumor or the RB, it always has to be this. This is the dominant manner, because what happens that, see that here, one copy is defective, but one copy is functional. It is producing functional RB. So here, it is a recessive gene. That's why in the heterozygous condition, the tumor is not observed. No tumor here. No tumor. Here you have that no tumor, and here you have the tumor. So why the gene by itself is recessive? That's why in heterozygous condition, its activity is recessed, it's recessive, because it is suppressed by the a normal gene. So only in cases that it is received in a dominant manner means that both the copies, both alleles are defective. Both copies of RB are defective to show the dominance or expression of the tumor. And here is a only case in there. So what is the difference between a tumor suppressor gene or this with the proto-oncogene? See that what is happening. Proto-oncogene also say, for example, A plus is normal and A minus. So A plus is for a tumor suppressor gene, say A plus is normal and say A is, I'm just writing, is high, say for example. And the possibilities are what you have, the A plus and the A plus and A plus, A high, and then you have an A high and A high, which means high hyperactive protein and constantly on. And this is, we are talking about the proto-oncogenes, so oncogenes. So what is happening? In this case, which is both is a normal, you do not see any cancer, no transformation in here. Here, one copy is high expression and you see cancer here. So you see cancer here, and of course you see cancer here as well. So here, the proto-oncogene is a dominant gene. So in the heterozygous condition, even if one is normal, it is dominant over that. And in a heterozygous condition, it is also causing cancer. And homozygous condition, obviously, it will cause cancer. Whereas, as the RB is a recessive gene, in the heterozygous condition, if one copy is effective, it will not cause a cancer. Only the dominant form is its both the alleles are in the non-functional form. And if you just dissect this out, it will make sense, because remember that the proto-oncogenes are constitutively off. That needs to be turned on. And if they are on in one allele, that will produce the oncoprotein. Whereas these genes are constantly on. And if one gene can produce the protein product, that will suppress the tumor. Focus on that just to make sure that you understand that what we mean by a recessive gene, and it's inherited in a dominant manner. Both the copies of RB are defective to show the dominance, not in the heterozygous condition. So let's see that how the loss of function can happen. All right. So after that, we are looking at that a loss of heterozygosity. So here, what we are seeing that there are two different ways that the loss of heterozygosity can actually have this mutation. So here, what we are looking at, you have a normal allele and you have a mutant allele. So the mutant allele, think about that this is an RB gene. So this is a heterozygous condition, and this person have a normal allele, and then the heterozygous condition, as the gene is recessive, it is not producing any issues. Now the chromosome duplicates during the cell division, it will duplicate. So each of the chromosome will have the sister chromatids, and one blue and one yellow should go to the one pole, but there could be a missegregation, and this is a problem with the spindle and all those kind of things, rare, but happens, of course. So if a chromosome receives a less number of chromosome, it will essentially die. But sometimes what happens that the cells will tolerate an extra chromosome for a little while, and then there will be a random loss of that extra chromosome. And by chance, if it happens that if it's a heterozygous, that is mutant allele, that is fine. Heterozygous is not going to show anything. But if the two defective or the mutant allele goes to another, it's a homozygous for the mutant allele. See that right here, this is the dominant form where both of the alleles are recessive genes, and only in here, the tumor suppressor gene will have its effect. In the heterozygous condition, as the gene is recessive, it will not have its effect. So the tumor suppressor function is maintained here just by the protein product of this one gene. But here, none of the gene can produce the protein product to have the tumor suppressor function. So that is the loss of function. So the mitotic recombination is, on the other hand, it happens all the time. It's discovered many, many years ago. A lot of people do not get that mitotic recombination is a fact. discovered in Drosophila probably 90 years back, 95 years back, almost a century back. And it's a common thing. Remember that mitotic recombination has nothing to do with the meiotic crossing over. The meiotic crossing over that happens in the formation of the gametes in the meiotic division, that happens only in the germ cell line while producing sperm and the oocytes or the gametes. So the meiotic recombination is an entirely different event. Mitotic recombination happens randomly in all cells, just randomly, but it is not controlled at all, just all the time. So here what we are seeing that a normal heterozygous condition, and here you have the chromosome duplication just before it goes into the mitosis. So you have the sister chromatids. And then in between the sister chromatids, just like crossing over, but this is not crossing over. We call this crossing over is a word that we use for the meiotic cell division for the germ cell line to produce the gametes. Here it's called the mitotic recombination. And you can see that the chromosomes, equal number of the copies of the chromosomes have been changed within this chromosome. And this happens more often into the chromosome pairs. So think about that. This is chromosome 4, this is chromosome 4. An equal amount of chromosome 4, one arm breaks and recombine. That is more common. But sometimes it happens between the chromosomes like 8, 14 as well in the Burkitt's lymphoma. So this happens. So this is also a case of mitotic recombination. Of course, that's how it leads to, which is very abnormal. So in this case, what happens that the one of the cases that the homozygous for the normal allele, this was originally it was this cell was a carrier, so one allele was defective. But after the mitotic recombination, one cell becomes both the alleles are now normal, whereas here both the alleles become defective, and this is homozygous for the mutant allele. So these are the way that the loss of heterozygosity can happen: mitotic recombination and missegregation. So again, the very straightforward answer that how the loss of heterozygosity can happen. Say which of the following statement is true. So loss of heterozygosity, say for example, can occur as a result of chromosome missegregation during the cell division. That is correct. Can occur as a result of recombination between homologous chromosomes. That is correct. Between the homologous chromosomes, recombination can contribute to transformed phenotype if it happens to a tumor suppressor gene. And now this is where people get confused. So remember that we are talking about the tumor suppressor gene, and this is missegregation and the mitotic recombination in event. Any of the tumor suppressor gene, if we mention right here, say for example, your 5, 6, 7, any of these genes, all of these mutant tumor suppressor genes, due to missegregation, due to the loss of heterozygosity, missegregation, or mitotic recombination, can lead to the cancer. So if we just have a statement, say for example, this is a P16 RB, then we are going to see at a P53, we are going to see. And if we just have a statement like this, that loss of heterozygosity can contribute to the transformed phenotype if it happens to, say for example, a P53 gene. So see that we talked about P53. P53 is a tumor suppressor gene. So that statement will be correct. If we say that can contribute to the transformed phenotype if it happens to an RB, yes, that can happen because RB is a tumor suppressor gene. So if we just have that statement for any of the tumor suppressor gene that we know the RB, P16, P53, just 3 of them is good enough. That this answer choice will be correct. A lot of people actually do not choose that answer because they think that only these 2 answers, missegregation and the mitotic recombination, is correct. But remember that these 2 essentially affecting all the tumor suppressor gene. That just remember the 3 most important one: RB, P53, maybe P16, you remember. But again, out of that, P53 and RB, these 2 will be the most important one as an example of that. So definitely remember those. All right, so that's type of question we can ask. All right, so after that, we are looking at some of the cell cycle control proteins. This is your class 5 cell cycle, so we'll go 5, 6, 7. So here, what you are seeing that the cyclin D and the CDK4, they are associated with a protein called P16. And the P16 is an inhibitor of the cyclin D CDK4. And what happened that when P16 is essentially removed from the cyclin D CDK4, the cyclin D CDK4 become active. And now the cyclin D CDK4 phosphorylates RB that release the E2F transcription factor. And now the E2F will essentially allow the cells to go to the mid to late to G1. So the cell will synthesize the DNA and will proceed towards the growth. So what happens here? P16 is a tumor suppressor gene. RB is a tumor suppressor gene. The P16 is on all the time to inhibit cyclin D CDK4. Only when it gets a signal, this will be released for the cyclin D CDK4 to activate. RB is bound to E2F all the time. When only cyclin D CDK4 is active under the correct condition, it will phosphorylate the RB. Now let's see that. So the RB is always on, P16 is always on. That's a normal thing. But think about that if you just lose that. So this is a defective. So suppose this becomes a defective. It cannot bind to cyclin D CDK4, or RB becomes a defective. It cannot bind to the E2F. What will essentially happen? That will lead to the cancer. So let's take a look at the next slide. So loss of P16, that will essentially cause a cancer. Loss of RB, that will lead to the cancer. So we just can ask this way, that which of the following proteins belong to the control system that regulates the G1S? So remember that which G1S transition, that is the important one. So you already know that. So mid to late G1, and this will lead to G1S, because you already know that the G1S transition. So because the synthesis phase is after the G1. So what is happening? That if we just ask the question that which of the following protein belong to the control system that regulates late G1 to S phase transition, often called as a start or the checkpoint. So remember that there were several checkpoints that we talked about with the anaphase promoting complex, mitotic checkpoint, G2 checkpoint, and this is essentially, this is your G1, this is your S, and this is your start checkpoint, which is about 3/4 up in the G1 phase, late G1 phase, and that's called the start. So if we just ask that which of the following protein belong to the control system that regulate late G1 to S phase checkpoint, often called the start checkpoints, your answer, if we just say, well, let's say CDK, if we say that CDK4, if we say cyclin D, if we say RB, P16, all of the proteins, essentially not E2F, E2F is the transcription factor, all of these proteins are responsible for this. Remember that the answer is not asking you that which is the tumor suppressor protein. Yeah, tumor suppressor protein activate and inactivate and activate this. The RB is phosphorylated and inactivated, but the cyclin D CDK4 is absolutely an integral part. If they do not phosphorylate the RB, it will not be activated. So this is a cell cycle control protein as well. So remember that along with P16 and RB. So again, repeating that, say, for example, the G1S transition on the checkpoint, CDK4, cyclin D, RB, P16, all of them are the part of that system as well. So if we just look at this, so just remember that right here, where is the start? So this is the start. This is essentially the start or the control checkpoint. And see that cyclin D, CDK4, right here, of course, the other one along with that. So this is an essential control, but how this works with that along with that, you have RB along with this, and then you have P16 along with this as well. So our P16, CDK4, cyclin D, and RB, all of these are essentially the part of it as well. So sometimes if we just don't have the P16 and say cyclin D, CDK4, and RB, that would be correct answer as well. So do not think that if we just don't have one of the component like a P16 is missing, it is a trick question. Cyclin D, CDK4, and RB, if these three choices are there, then all of these are correct answer for the control system at the checkpoint. That will help. Yep, that's pretty much all of it. And so the question is again, you do not have to memorize anything from here. The only reason that I just put it in here, this just again emphasizes the role of p53, and just again reminding you that where the p53 is essentially blocking the cell cycle. So you can see that there are 4 DNA damage checkpoints, and if there are DNA damage, the p53 is induced, and it will have an immediately rapid block at the entry of the S phase, at the back of the S phase, entry to the mitosis, and also in the G1 phase. So 4 DNA damage checkpoint, but no direct question from this one, just reminding you from there. So how p53 essentially works, this is more important, which describes p53. So the p53 is stabilized by the DNA damage, and it is a transcription factor. So it is stabilized and induces the synthesis of p21 CYP. And that will bind to the cyclin CDKs in certain ways. That, remember that it could be many different ones. That's why it is not specific. It can bind to the cyclin DCDK4/6. Here it is cyclin EA, cyclin A, cyclin AB. So it can bind to any one of them. But essentially the CYP is the binding inhibitory protein for the cyclin CDK, which was arrest in the G1, G2, 4 different checkpoints. So the typical question that we can ask is that which of the following statement correctly describes p53? So remember that the activation of p53 leads to the inhibition of the cyclin CDK complexes, inhibition of the cyclin CDK complexes. That's the correct answer. On the other hand, if we just see some of the distractors, it is a membrane receptor that binds to a growth factor like EGF. No, it is a soluble protein produced and present in the nucleus. It is not an EGF, not a growth factor, nothing. Cell division stops until p53 binds to DNA, repairs the damage. No, the p53, it is activated by the DNA. So ATM, ATRs are the DNA binding proteins, which stabilizes the p53. p53 is stabilized by DNA damage, but it is not by itself a DNA binding protein. Remember that. On the other hand, if we just say that p53 is an example of an oncogene, remember that it's a tumor suppressor gene. It is not an oncogene. So just remember that different. On the other hand, say, for example, if we say the p53 bind to pre-replication complexes, promoting transition from G1 to S phase. Well, this doesn't promote G1 to S phase. It doesn't bind to any pre-replication complex. Its function is only to induce synthesis of inhibitors of cell cycle control. So this is the CYP, p21 CYP, which arrest the cell cycle and also produce an apoptotic proteins. Transcription factor that regulates transcription of this. So this is the type of question that we can ask. So then the next one that what we are seeing that this is one of the important slides that essentially shows the different things. Here you have a damaged DNA. And the damaged DNA, the ATM, there are 2 proteins, ATM and ATR. If you see that where they are, this is right here, ATM, ATR. These ATM and ATRs bind to the DNA, and there are several different types of things can go on. So this CDC25A, this is essentially an activator of the cyclin CDK. Remember that. It is an activator of cyclin CDK. The ATMs can induce or phosphorylate. It is a kinase as well. The ATM or ATR, both are kinases. It phosphorylates the CHEK2. And when CHEK2 phosphorylates CDC25A, this is the activator of the cell cycle or the cyclin CDKs. This undergoes the proteasomal degradation, and this is a rapid G1S block. This is one pathway. So let's see that where we are seeing right here. If you look at this, see that right here, CDC25A, this is, I will just point it down. So the CDC25A is an activator of this. And see, check 1 to 2 essentially blocks this or destroy the CDC25A so that this cannot be activated as well. And also see that ATM, ATR also activate p53, p21 CYP. So this is a double-pronged control. So one is destruction of an activator of the cell cycle, and another is producing an inhibitor of the cell cycle. And this is, again, this is an activator. Remember that this is an activator of cyclin and the CDKs. So let's go back in here. So let's see the main thing in here. Now, what happens that we will start from here and we'll come back in here. So in the cell, the p53 is constantly produced and its half-life is about 30 minutes. Within 30 minutes, it binds to a protein called MDM2, and it is labeled for proteasomal degradation. So p53 in a normal cell is produced, degraded. But what happens that when there is DNA damage observed by ATM-ATR binding, this kinase activates and it phosphorylates the p53, it stabilizes the p53. And this will lead to the transcription activation, and see that what is happening. It will induce synthesis of DNA repair proteins. It will have a sustained G1, G2 arrest through the P21 CYP production, and it will also induce production of proteins that leads to controlled programmed cell death or apoptosis. So these are the three outcome or function of the p53. It's a transcription activation. Lots of gene can be activated by this. Now let's see that another way that in cancer cells, what happens. So in cancer cells, what happens that the MDM2's function is to bind to p53 and degrade it. The ATM, in addition to directly stabilize p53, it also phosphorylates the MDM2. So when it phosphorylates the MDM cannot bind to p53 and the pool of p53 actually increase. That means that p53 pool increase means it's stabilized. So apoptosis, sustained G1, G2 arrest, and DNA repair, all is going to take place. I'll explain hyperphosphorylation. Another thing also happened. If the cell actually understand that the cell is undergoing an uncontrolled cell growth or an oncogenic signaling is going on, cell have a sense for doing that as well. And that's why you need those 3 to 7 multiple mutations. If this any oncogenic signaling is going on, it produces a protein called P14ARF. The P14ARF also sequester or binds to MDM2. And that means that MDM2 will not be able to bind to p53 and its pool will increase. The same thing. So MDM2 by hyperphosphorylation will unable to bind p53. MDM2 by P14ARF, this will also help the MDM2 not to bind. So all these measures are anti-cancer measures. Pool of p53 increase. But what happens, sometimes a mutated P14ARF, sometimes they become inactive and sequestered in the nucleus. So this P14ARF, this protein is produced due to the oncogenic signaling, but due to a mutation, this is essentially a tumor suppressor protein. So because it is promoting the tumor suppressing, and this is sequestered in the nucleus. It can't bind to MDM2 in an oncogenic signaling, DNA damage, uncontrolled cell division. So it bind to P53 and degrade them. So essentially, that's basically the MDM. So the MDM can bind to P53 and do this. Now, let's see that this is also what can happen. We see that the MDM2 is hyperphosphorylated in the tumor cells. So a lot of people asking that, isn't that a controversial statement, or it is just not saying the other thing? If MDM2 is hyperphosphorylated, it is increasing the P53 and it is stabilizing the P53. So why MDM2 is hyperphosphorylated in the cells? Remember that what is happening in here, and this is where that the 3 to 7 mutations come into play. There are many other mutations in different places that is essentially telling the cell for uncontrolled cell division. The cell is getting an oncogenic signaling, ATM may be activated, MDM2 is hyperphosphorylated, and suppose P53 pool is increasing. But suppose here, the protein that it is producing P21 CYP, suppose that protein is mutated. So even if P53 is high, even if this is high, even this is working, the protein that is the inhibitor of the G1, G2 arrest is mutated. So even if this is high, it cannot control it. Suppose some of the apoptotic proteins are mutated. Suppose some of the DNA repair proteins are mutated. In that cases, high P53, the cell is trying its best to increase the pool of P53 but the effect is futile because the mutation is somewhere else. Like, say, for example, some proteins which is responsible for G1, G2 arrest. Some mutations are in the DNA repair protein. Some proteins are in the some mutations in the apoptotic protein, so the cell cannot die. So even though you have a high amount of this, all working towards this, the cell cannot control the cancer essentially. So you will see that I will just post somebody asked a question, and I will post a discussion post that essentially what happens, a very common thing that actually happens, that how you stain the tumor cells. So when you take a biopsy, you actually stain with their P53. It's called a P53 staining. So in tumor cells, you see a high amount of P53. It's a standard assay. And the question is that you always see that when pool of P53 increase, this is essentially happening. Why P53 is increased in that? Remember that the P53 increase does not mean that it is able to control, because there are 3 to 7 genetic accidents that is elsewhere, and that can lead to even with that. But the P53 is always in a higher amount in these cells. So that explains that. So question that we can ask from here, say, for example, we will ask a question from P14 ARF. So this is how the question may be formed. Cell suffered from a mutation in the RAS oncogene, leading to the cell loss of cell control of cell division. The event induced synthesis of a protein as P14 ARF. Remember that RAS lost the control, and that is an oncogenic signaling. The cell can sense that, and it will produce the ARF, which binds to the protein of MDM2 and renders it inactive, so that it cannot no longer bind to the P53, and that will lead to the pool of P53. So in that sense, we will ask after that which of the following is the correct designation for the P14 ARF. Remember that P14 is essentially binding to MDM2, increasing P53, whose function is to check the cell division. So the P14 is also a tumor suppressor gene. But this is a tumor suppressor protein that most of the tumor suppressor protein are produced all the time. And they are, when they are inactive, they have a problem. The P14 ARF, being a tumor suppressor gene, is produced only specific to oncogenic signaling. But remember that it's a tumor suppressor gene because it is increasing a tumor suppressor protein. All right. So here you can see that P53 is various. 70% of the cancers can lead to these P53. So see that what happens, that P53 sometimes are mutated, their levels are high, but the mutations make this P53 staining possible with the high amount in the cell. But the P53 is not able to do its transcription because you can have a mutation in these regions. So if you carefully look at that, most of these hotspot or the highest mutations, they're all arginine 282, 273, 249, 248, and 175. So think about that, why they are all arginine. These arginines are all positively charged, and see that they are all in the DNA binding domains. The DNA is negatively charged, and these are the positively charged amino acid. So these are the DNA binding domains. If you mutate them, they lose the ability to bind to the DNA and cannot have the effect of the transcription factor. So what it will lead to, essentially coming together, it will not be able to produce the protein, apoptotic protein, will not be able to produce the P21 CYP, will not be able to induce the synthesis of the DNA repair protein, because the mutation does not allow the transcription factor to bind to the DNA properly and induce that. Its level may be high. Its level may be high because of the MDM2, ARF, phosphorylation. Its level is high, but it's an ineffective or P53 in the cell. Sometimes that is also observed. The cell is staining for high amount of P53, but it's unable to do its function as well. All these different combination of scenarios are possible. All right. So after that, we are looking at, this is the class 6 DNA repair gene. And these are the famous We call this BRCA1. Sometimes we call it BRCA1 or BRCA2 or BRCA1, BRCA2. And these are implicated in hereditary breast cancer gene. These genes actually produce proteins which are enzymes for the double-stranded DNA break repair. And when these genes are essentially lost, there is a loss of genome integrity, unregulated transcription, unregulated protein ubiquitination, and ultimately lead to the cell cycle control. Very straightforward question. We can just ask that straightforward, which of the following genes code for DNA repair proteins often implicated in hereditary breast cancer? BRCA1, BRCA2 is the answer. Not P53, not RAS, not MYC. Remember that. So very straightforward questions. And after that, what I did is after that, you have these current and conventional treatments, clinical trials. And again, this is not required. And these are some of the clinical trials and future perspectives. And many of you may be interested in some of these very, very promising areas. All right. So that's pretty much it, I guess.